As we have seen in previous modules, with modern technologies in the image processing, it's possible to create a digital 3D model of a patient's tissues. These computational procedures, finalized to visualization and quantification, can support clinicians in disease diagnosing, definition of biomarkers related to pathologies, and to monitor the progression of diseases. In this module we will see examples of organ segmentation and volume quantification. You can practice using 3D Slicer software previously mentioned to obtain our same results. For this task you can use the demo images provided by 3D Slicer. As shown in this slide, after starting 3D Slicer software, we can select the menu, Sample Data, as indicated here by the red arrow. A number of demo images can be selected and among them some are related to lung CT. Let's now select the CT images with labeled Demo Chest CT, as indicated by the red arrow. The images of the demo CT scan are then displayed in the horizontal, sagittal and coronal view. You can visualize different images in the CT scan using the cursors indicated by the green arrow. We are going to see how to segment and then analyze lung tissue from the demo chest CT images. It would be impossible to manually segment the contour of the lungs in all the horizontal images. Using 3D Slicer it is possible to accomplish this task with automatic procedures that require minimal manual operations. The automatic segmentation we are going to obtain is based on the region growing techniques, that is based on the difference in radiodensity between the lungs and the surrounding tissues. To launch the segmentation algorithm we select, on the menu, Sample Data, the submenu Chest Imaging Platform. It is then possible to select the submenu Lung CT Segmenter, as shown by the red arrow. Selecting this submenu we obtain the result shown in the next slide. The software displays the images with intensities used for lung analysis. Let's now press the Start button as indicated by the red arrow. As a first step, we need to place 3 seed points in both lungs in the axial plane, as indicated by the red arrows. Moving the cursor and clicking on the lung tissue. The points are labeled with a number. After the selection of the last seed point, another view is displayed, as shown in the next slide. Then we need to place a seed point in the trachea, as shown by the red arrow. Once this point is selected, the software displays the following view. Now we need to click on the Apply button to launch the automatic segmentation. The result is shown in the next slide. In a matter of seconds, the 3D model of the patient's lungs is computed and shown in the three-dimensional view. This model can be saved by clicking on the Save button in the upper left of the window. Removing the seed points, the lung model shows in detail the 2 lungs and the trachea. By selecting the Maximize view, on the 3D view located in the upper right corner, we can better visualize the result of the 3D segmentation process to evaluate it, as shown in the next slide. We can appreciate the detail of the reconstruction, that is based on data automatically segmented from CT image. It's also possible to save the 3D model as a file. The standard file for 3-dimensional objects is the stereolithography format or STL, as shown in the next slide. By selecting the Export to Files module we can select the file format as STL, as shown here by the red arrow. We can then press the Export button to save the file in our computer. Solid numerical models in STL format can be visualized and processed using different visualization softwares. As shown here we have loaded the STL file in the software ParaView. The same 3-dimensional model we have visualized in 3D Slicer can be visualized with the software ParaView. The surface represented is based on a set of data that contains the coordinates of the vertices of a number of triangles. We can visualize these triangles changing the surface representation indicated by the red arrow, with the surface with edges representation as shown in the next slide. These triangles, with their orientation, are used to visualize the surface. In summary, we have seen how to automatically segment an organ from a CT image sequence, how it is possible to visualize it in 3 dimensions, how to save the model in a file, and finally how to visualize the model with other software tools. Thank you for listening to this lesson. Please go to the next lesson of this module to learn on the use of artificial intelligence tools in 3D Slicer to process radiological images.